All right, so I hope you enjoyed that activity and make sure you understood what we're actually looking at here. All right. Oh, let's go back. Okay, so now what we want to do, we finished looking at transcription, which is the first part of making a protein. Now what we want to do is look at translation, which is the second part of making a protein. All right. What we want to do is we want to use this RNA molecule that we just made. We want to use that to make a protein. Now that usually happens with the help of a ribosome. Okay, so now I think that we may go back to this a little bit later. Let's build the protein first, and then we can kind of think about codons and stuff like that. Essentially, what it is that RNA, the way we use the RNA molecule is that we read it in sets of 3 letters. Okay, that's basically what it means. So 3 letters, so 3 bases of RNA is termed a codon. It's just another bit of terminology. Don't worry too much about it. Right, so this is what we want to do. This is what we want to do to make a protein. Here is our RNA, and then these things here, they each carry 1 amino acid. So what we want to do is we want to make a protein using this RNA as a template. Okay. Now I've got like an animation for you to watch, so it's okay. This just shows you step by step what kind of happens, but I think it's probably better that we build one, and then yeah, might be better. And then you'll understand these pictures a little bit better. Here is another picture of what we're trying to achieve, or another animation of what we're trying to achieve. But yeah, let's build one. Oh, I don't want to watch that. Okay. So here we go. I'm going to do this in outer presenter view so you can see exactly, I can teach you exactly how I want to. Oh, where's my camera gone? Oh no. Here I am, back again. I've temporarily disappeared to my other screen. Let me just make this as small as possible for you so you can actually see what I'm doing here, because this is the important part. Right. So this is what is called translation, because we're actually going from nucleic acid language. Translation is going from nucleotide language to amino acid language. So let's have a look at this first. First of all, I want to show you this molecule here. See this guy? Okay. This guy is called a transfer RNA, and he is floating around in the cell, just hanging around waiting to be used. Now what happens is that this transfer RNA locks in. So this guy here, this transfer RNA, is carrying, say, a type of amino acid. Now there's 20 types of amino acids. I don't want you to worry too much about what amino acid this one corresponds to. Let's just say that this is an amino acid. So this is amino acid number 1, okay? Right, so here we go. So he just floats around and then comes back here, and then in the ribosome locks into place, okay? Because they match, okay? U is always going to U is married to A is married to U, C is married to G. So this guy here will only lock in that spot. He can't lock here. It doesn't fit. It's not right. Okay? So see how they match? You have to have the matching base pairs in order for this guy to lock. So this guy fits into here. Yay, awesome. He is the start of our protein, right? Now, what we want to do is we want to build an amino acid chain, okay? We want to build an amino acid chain, which means that we have one amino acid. Hooray! Now, let's find the other one that locks in. So what goes with GUA? GUA. I think it's this one. So this one should match this guy here. C is married to G, A is married to U, and U is married to A. Yeah, that's the correct one. So that is the only one that can go next. Right, now what happens in the ribosome is this. Let me just ungroup these guys. I'm just going to show you. What happens in the ribosome is that this amino acid, let me just ungroup this one too. I just need to ungroup all of them, sorry. Right. In the ribosome, this amino acid locks into this amino acid, okay? And then this and this and this and this floats away. So now we have amino acid 1 joined with amino acid 2. Yay! All right, so let's add one more amino acid and then I'll let you finish it off and finish your own protein. Here we go. Let's see which one locks in next. I think it's this one. Please tell me if I'm wrong. C goes with G, G goes with C, and A goes with this one. That one is the one next to lock in the chain because it matches. Right, so this one is going to be our amino acid 3. So the ribosome grabs these two guys, locks them in. They disappear, and now we have 1, 2, 3. Can you see how we are gradually building our amino acid chain? That's basically how it works. So I'll let you finish off your amino acid chain, but essentially it's like this. Let me just show you a picture. Oops. Here we go. Look at this. This is making our RNA molecule. Pretty cool, hey? There we go. We're making it. There we go. We nearly made it. They all match up. And now we made our RNA, and now we go to the cytoplasm, and now we're going to make a amino acid chain. Look at this. Yay! Look. Yay! See? That's exactly what I did on my activity. So I want you to do that to understand, yes? It's about understanding stuff. Here is another picture that shows you exactly how proteins, this is from, see, DNA, RNA. These are the little transfer RNAs. And then we end up with a beautiful amino acid chain. Happy days. So as you can see here, GUA carries this amino acid. CUC carry this amino acid. So they're all different. And remember, when you have a chain of amino acids, they fold and they make a protein. Remember the 4 levels of protein structure? Yeah. Yay!